Good afternoon, everybody. If I can just ask you to take your seats so that we can start this session. Okay, good afternoon, and thank you very much for the invitation to moderate this fantastic session. My name is Bauermeister, and today I'm going to make a few assumptions. One, that everyone is a little tired, and two, that you've read all the biographies, because I'm not going to say much about our speakers. They can introduce themselves. So this session today is called From Cohorts to Data Validation. And it's really calling to attention the complexities of cohort and participant data, and large-scale data management, and also analyses to prepare data sets for downstream health and biomedical research. And we have three fantastic speakers today who are extremely experienced in what they do. They are also representing very diverse communities of our global community. So I'd like to welcome to the stage Professor Nicola Mulder from the University of Cape Town, who is going to speak about cohort data management and interpretation in an African setting. I was also supposed to say, don't forget the Q&A and the pigeonhole throughout the session. I should have said that first.
Hi, everyone. I hope you are still awake, especially after lunch. And thanks to the organizers for the invitation to speak. So as Sarah mentioned, I'm going to be talking about the African setting. And she's already given the context for this particular session about the complexities of data when you're looking at cohorts. And of course, this if you look at the entire data ecosystem, the whole data life cycle, there are a lot of different pieces, which I'm sure all of you know about these individual pieces. But it really starts from the participant recruitment and the data that gets collected there in terms of the informed consent. And Nikki Tiffin spoke earlier about their electronic version of informed consent, which makes life so much easier when you're trying to tease apart for the data later, the data sharing, who consented to what in terms of the data sharing. Of course, the clinical data can be from electronic health records, from CRF case report forms, from wearables. But then I think a lot of people forget about the metadata. Metadata associated with the cohort study. What is the study about? What is the population group you're working on? And then the metadata associated with all the different samples. These are all key pieces of the data kind of ecosystem. And then there's the data management, the boring stuff of data transfer, storage, and then sharing, and the security that we need for that. So encryption, for example, when you're working with human data. And then, of course, the actual part that comes down to preparing the data for that biomedical research and that health translation. And that's really getting the data at to a top level of quality, but also to make the data usable. So there's VCF files. There's all sorts of different file formats, not necessarily useful for the interpretation of the data. You need to convert that into something useful. And then you need to have the right tools. And I'm going to talk a little bit mostly about this bottom section of the data. And I know that the other two speakers, I presume the other two speakers, are going to be talking about single countries. Here I'm going to be looking at the multi-country, multi-site perspective. So Africa, one continent, yes. Many people talk of Africa like it's one country. So everybody talks about African data, African frequencies, African this. Africa is 54, 55, depending on your politics, countries with an extreme diversity in climate, in geography, in wealth, and exposure to infectious diseases. And all of these factors have shaped the genomes of African populations. For example, all the populations that have been exposed to malaria in the malaria belt, they have very distinct SNPs that protect against malaria but predispose to other diseases. So when you're looking at multi-country studies, you need to think about the genetic diversity, which we've all heard about many times in Africa, but also varying levels of governance in terms of how mature they are in managing data and data governance. So the different data protection laws that are in these countries, and different levels of scientific infrastructure and capacity to do research. So that's that equity, having collaborations across multi-country sites that are at different levels of maturity. You need to be really careful that you don't have dominance by specific groups. There needs to be complete equity in that. So all of these impact multi-country and multi-site cohort studies. So I've been working in 2 different consortia that have multi-country sites, and these are all at both projects on Africa. So H3 Africa, I think many people know about, multiple different sites, multiple different diseases, and more than 30 different countries. So I was running H3 Bionet in that, which was managing all the data infrastructure. The other project that I've been involved in more recently is the Harnessing Data Science for Health Innovation in Africa, the DSI Africa. And this is a project, multi-country project across Africa that's using secondary data. So it's whereas H3 Africa was generating the data, DSI Africa is using existing data sets, but different data sets. So it's not just genomics focus, climate, environment, images, all sorts of things, and using data science technologies. So in that, we've got an open data science platform that we're also using to manage the data. So the number of advantages of multi-country studies, of course, you have migration across borders, particularly in Africa, where somebody else set our borders. So you don't have neat population groups and therefore neat genetic groups within each different country. You have population admixture because people move. And so these multi-country studies allow you to capture both the diversity but also the similarities across different borders. Of course, with COVID, we know that infectious diseases don't know boundaries, and so it was really important to have studies across different countries. And then, of course, there's the sample size, the enabling of comparisons across populations, having replication sites, shared resources and collaboration, particularly important in low-resource settings. But the challenges, you've heard about all the challenges, particularly in the LMIC session today, and I'm sure you don't want to hear about more, but I'm probably going to introduce more of those challenges. So, of course, there's heterogeneity in data collection because you've got multiple different sites, and unless there's a lot of standardization up front, there's going to be heterogeneity. You have to think about the large-scale data management, moving data across borders. And then of all the things we've heard about already, the uniqueness of African genomes, are the tools correct for analyzing the data? And is the data in a format, there's so little African data, is it all findable and in the right format to get value out of that data? And then also considering the diversity, what is the level of granularity of diversity you need to go to where there's a health implication? So some of the solutions I'm going to talk about are the data harmonization across sites, how we can do centralized or federated analysis. Some of the tools have been working with the Human Pangenome Reference Consortium to build a better reference graph. And some of the other tools we have worked on. So I'll talk about some of those. But firstly, the data harmonization. So we know this prospective harmonization, obviously that's the best. You set the standards beforehand, and then you can control how the data are collected. But in H3 Africa, and of course DSI Africa, where these are all secondary data sets, that was just not possible. And so you have to think about retrospective harmonization algorithms. So for prospective harmonization, through the lessons of H3 Africa, the H3 Africa Phenotype Harmonization Working Group actually put together a set of case report forms for the next round of H3 Africa projects. So this was done by taking all the case report forms from the existing projects, seeing where the overlaps were, mapping those to Phoenix, for example, or standard ways of collecting data, and then creating new protocols in REDCap. And these were then externally reviewed, mapped to ontologies, and now we've created a resource for those. So we have standard CRF modules for adults and pediatrics, for the standard demographics, for environmental lifestyle factors, and then for specific diseases. So we've got 11 data collection toolkits, a number of different ones, and we've also had to adapt these to the African setting. Dietary questions are going to be different in the US compared to in rural Africa. And for each of these, we've actually created REDCap templates. We have Excel spreadsheets. We have PDF forms. And these are all openly available on our website and also on a GitHub repository. And the second round of H3 Africa projects actually use these standard case report forms. So this is very nice if you're doing a prospective study. But for retrospective data harmonization, we had to take all the data elements that were being collected and look at the overlaps. And so the heat map shows for a set of data elements which of the projects were collecting those. And then when we were doing data harmonization, we developed algorithms that actually then had to map to the lowest common denominator. So if one answered yes or no, and others had a particular value for that, you couldn't take that value. You had to go to the yes/no answer. But even these algorithms still require manual input, as anybody who's done data harmonization would know. In the DSI Africa Consortium, we've also obviously hit upon the data harmonization requirements. And so we've developed some tools that take the metadata and the data and then do some processing, mapping to models, and then provide a clean output. So one of the DSI Africa projects, the HEAT project, they actually developed this algorithm where you take you input your data set variables as a CSV file, you input your code book, and then other data if you need, and then it actually runs large language models and provides some recommendations for the mapping to the code book. And so you end up with these data set variables mapped to this code book. So what we've done in Awazi is to take that and put it into a nice web interface that you can upload your data and then make the selections if you want to have manual input. And it's containerized, so anybody can take this and use this tool. So in this way, we've now got all the projects to at least harmonize their metadata, and then we created a REDCap form for them to upload their metadata into, and that then makes sure that, because we want all the data to be findable, so we now have put that into, it uses an API to call it into our catalog. And so we've got the Awazi catalog that now lists all the data sets that have put their metadata in DSI, the types of data sets. Some are related to pollution, some to genomics data, some to clinical data. So at least you can find the data sets, and then we also include the data use permissions through the data use ontology mapping. So this is one where we've been trying to harmonize the metadata across multiple different studies. Of course, the next challenge is the more studies you include, the bigger the data sets. How do you manage those data sets? And so you've got to integrate with omics data, with the health data. If you look across African countries, there are very few countries that have really good quality electronic health record systems. And so you have to manage how you bring health data in across different sites. And of course, it's managing this cross-data border sharing. So in 2022, the African Union published their data policy framework. And at the time, there were very few countries that had proper data protection laws in. But in the last 2 years, a lot of the countries, up to 30 countries, have just implemented data protection laws. And some of them allow for sharing if the other country you're sharing with has such a policy in place. But it's not just the policies that are a barrier. There's also hesitation by the scientists to share data anyway, let alone across borders in these cross-country sites. So we're also looking at technical implementation. So how do we deal with federated versus centralized? We're using centralized, obviously, for the metadata, where it's just sort of what data are you collecting. But for the actual data, we're using GA4GH standards, so WES, DIRS, and Data Connect, and also the passports, to implement federated analysis. So we've tested in Mali, Uganda, and Cape Town, 3 different sites, also using Gen 3, which uses the GA4GH standards, to think about how we can do federated analysis without having to move the data. So the next section I'm going to talk about is how you manage the data in terms of getting it ready for actual interpretation. And this is just talking particularly about some of the tools. So if we think about how you get to health benefits, data is just data. It maybe can give you information, but it doesn't give you knowledge without that actual interpretation through a use of tools. So you heard about the imputation panel we have and the H-Africa array. We're also thinking about how we can use, for whole genome sequence data, how we can get a better reference genome. And then I'll talk about allele frequency and granularity. So for the African reference graph, we're working with the Human Pangenome Reference Consortium. We've got some long-read data that we've had sequenced from different countries, and we've put those together. We've generated a reference graph, and we're currently benchmarking that against for variant calling using different African data sets. We've had some success. One of our neuromuscular diseases cohorts in Cape Town has tested this, and they've got great results. So we're actually running a hackathon in October for people who want to, the African researchers who want to come and test their data. So after you've done your variant calling and you've done your analysis and your associations, you then want to think about how you can interpret the results. What is the right variant you're looking for? And in many cases, people use allele frequency data. So we've developed this African genome variation database. Now, it's very similar. In fact, it's kind of mirror to NOMAD. The reason we're doing this instead of putting our data in NOMAD is I'd love to have all the data in NOMAD, but there was resistance. There was a request to do so. There was resistance from the African PIs to do so because of one is American and one is African. But also, NOMAD, again, calls it African, and so there's one frequency for the whole kind of continent, and we needed frequency at a more granular level. So we also then had this decision to make of how do we cluster the populations to calculate these frequencies and at what level of granularity is not too granular that it's sensitive in terms of it gets to a particular population or a particular tribal group. So a population can be in many different countries, and a country can have multiple different populations. A study can have multiple different populations, or a population can be in multiple studies. So you can't just manually kind of group them by those criteria. You also need a minimal sample size to make the frequency of any value. And the sensitivity thing, you don't want to be so granular that you have identifiability. So if you look at, so this is data from a Nature paper that we used Nature Africa data for. You can see that the clusters don't, in many cases, they group by sort of region and country. But if you look at the kind of long oval there, you can see, for those of you who know, which you won't, that U is for Uganda. So UNB and UBS are two different populations from Uganda. So one country is separating quite significantly. So we can't just say manually, oh, we'll cluster every country, all the groups that are in West Africa by country. So we actually took the data that we were putting in AGVD and we clustered it both manually and then computationally using FST and various other criteria for determining how to assign clusters. And you can see that mostly it's fine, but in the red section there, you can see that some of the population separates out into different clusters. And in most cases, the frequencies are sort of similar. But there's this example, in this case, there's a very rare variant which is hardly found outside of Africa. In Africa, this is the bar chart for data from NOMAD. It's 0.0009 in African populations. If you look at the next plot, the first one is the computational clustering and the other one is the manual clustering. And you can see that even though it's a specific frequency in African populations, it's actually only found in one cluster in those African populations, not seen anywhere in any of the others. And the frequencies differ depending on whether you did manual clustering or computational clustering. And you can see why, because you're including different countries there. So it depends on the level that you need to go to in terms of making a clinical decision. Is it important that it's 0.002 versus 0.008 in terms of the decision you're going to make and therefore how you cluster the variants? So we actually present both in AGVD. We show both the manual clustering and the computational clustering so people can decide. And then when you look at the variants, you can actually see which countries are represented in each of these. We don't show the population groups, but we show the countries. So this is the example. So that was that was the clusters where that particular variant came from, and you can see why there was a different frequency. Even just sample numbers are going to make a difference. So we're leveraging AGVD for rare diseases and a number of rare diseases groups in South Africa, but also in the US, who are working with, for example, the Somali population in New York and various different places. They actually want to use this for more granular analysis of variant data. So if you think about the whole genome sequencing pipeline for rare variants, you filter to remove common variants, and then you identify, you use variant frequency to determine what you want. Now in AGVD, because we've got the trust of the African researchers to have the data, we still want to make it sort of a little bit protected. So anybody can use it, but at the moment we want you to register to use it. So anyone can register. So you don't have to pay, you don't have to, pass a test. But we just want to we just want to see who's using it for what, before people are, while they're still comfortable with sharing it. So we can't make big data sets available. So for the moment, we're just creating an API where you can actually filter your list of potential variants from a rare variant patient, and then it gives you a ranked list depending on what your criteria are. And then we also point to the Beacon network as well. But regional variation matters also for complex diseases. So actually, this is a study from Michelle Ramsey's group. So 2 studies I'm going to talk about from East Africa, where they identified a novel association for LDLC. And this variant was only pretty much absent from non-African populations. It had very low monoallele frequency in non-Africans. And if you look at the clustering in the AGVD, you'll see that East Africa is really low. Compared to the rest. And so suddenly you need to think about where's your patient coming from if you're going to have any action on this. Same with this other variant, also a study from an East Africa group. This is a novel association with diastolic blood pressure. In NOMAD, there's just one sort of bar for African, but when you look at the regional variation, there's distinct kind of dominance of this in West Africa and North and East Africa, but Central Southern Africa, it's very rare. Same with clinically important pharmacogenomic variants. So we took all the variants in PharmGKB level 1AB, and this is the plot shows the distribution of allele frequencies for different African subgroups, subpopulations. And you'll see that there's huge variation in terms of the allele frequency. There's some for this particular gene which are only found in East African populations. And then interesting, some for the same gene, the frequencies are very similar. But there are other genes where 2 SNPs from the same gene have very different allele frequency patterns across these different populations. So again, the importance of looking at the regional level variation. So just to sum up with some take-home messages, I think the basic answer, the long story short, is that management of data across multi-study, multi-country cohorts is extremely complex. And it's to do with both the heterogeneity, but also the governance. We need to think about central versus federated. We have many complexities in terms of also resources. We need to consider having the right tools, looking at the data at the right level of granularity, but ultimately we need more African data. So we've got a number of initiatives that are trying to generate more data, and Mark talked about the pilot project we have with PAGRA to sequence 25,000 African genomes, which we hope will significantly boost our data sets. So with that, I'd just like to acknowledge the H3 Africa, H3 Bionet, and Alwazi consortia, and the funding from the NIH. And thank you for your attention.
Very much, Nikki, for a great talk. Please, can I just remind you about the QR code? Please submit your questions, and in the Q&A session, we will go through those questions with the panel here up on stage. Our next speaker is Dr. Nahla Al-Afifi, and she is representing the Qatar Biobank study and is standing in for her colleague, Dr. Qafut. So looking at Qatar Biobank from cohort to clinics, validating data integrity across the healthcare spectrum. Welcome to the stand.
Hello, everyone. Sorry for the last-minute change between me and my friend, Dr. Fatima, as she didn't feel well. So I will speak on behalf of her on validating data integrity across the healthcare spectrum. As we are in the Biobank, Qatar, if you know that, it is a small peninsula within the Middle East and Gulf region, and with population around 2.5 million person, and around 10% of the population is Qatari, and the rest are expats coming to Qatar for working. So Qatar Biobank is the central resource for biobanking. It is The vision is mainly, it is established in 2012 by collaboration between the Ministry of Public Health, Hamad Medical Corporation, and on the support of our expertise from England, from UK, from Imperial College. So then Qatar, from the beginning, the vision of the Qatar Biobank is to act as a national center for biological samples and information to enable research toward the discovery and development of the new healthcare intervention. And of course, our mission is that we are concentrating as a population health insight, precision health, and global collaboration with to advance personalized healthcare, ensuring benefits of Qatar region and international community. So Qatar Biobank is, as I mentioned, is allocated in Hamad Medical Corporation, where the center for the medical hospitals, and it has to have where we collect the participant come to the biobank, we collect the samples and data, then we send the samples to the hospital as it is accredited hospital for getting the results, and send the samples also, DNA samples to Sidra Medicine for the whole genome sequence. And then for omic data, we send it, we are working in collaboration with the anti-doping lab for metabolomic and proteomics with Weill Cornell Medicine. So as I mentioned that since 2012, we started the population-based biobank. We reach, after 10 years, we arrived that we have more than 40,000 participants within Qatar Biobank. This is the main pilot study, and after every 5 years, the participants come for revisit. Now we have more than 10,000 participants are coming for second visit. Then we have initiated another initiatives like for our participant that to link our data with the electronic medical records, which is centralized in Qatar using the CERNL. And then we have the tissue biobank, establishing a tissue biobank. We are first phase of tissue biobank with collaboration with the hospital, Central Hospital Pathology Lab, where we are concentrating on cancer breast and cancer colon. Then we have the Qatar Genome Program, which was established and incubated within the biobank for sequencing our participants now with the new establishing of QPHI, which Qatar Precision Health Institute. Both of us are under this umbrella. Then we have the birth cohort study, which is called cubic study, a Qatar birth cohort study, where recruiting the mother, father, and pregnant woman, the pregnant woman and the father, and also follow the outcome of the baby for 6 years. Then we have the MRI visit. I will mention it in detail that we do the brain and whole body. And then we started now a specific disease cohorts where the researchers approach the biobank, asking them to support and collect for them the data and samples. So we have a cardiovascular biorepository, we have COVID biorepository, and cystic fibrosis biorepository, according to the request of the researchers. The wealth of the data which is collected in the biobank is huge. As I mentioned, that our model is the participant coming to the biobank. We are not a hospital. They come voluntarily, we register them. We have a visit of 3 hours, they come. We, after everything is electronically collected from the consent, then we have a questionnaire, a comprehensive questionnaire about their lifestyle, health, and we have nurse questionnaire with contraindication, and we have dietary questionnaire. So the questionnaire is around 1.5 hours nearly with all of this. Then after that, we start the clinical measurements within the biobank. We collect the data from ultrasound, carotid ultrasound, blood pressure, of course, height, weight, and all demographic data within the biobank. We do stress test. And then at the end of the visit, there is another questionnaire about if they are eligible for MRI. If they are eligible for MRI, they will come for second visit, where we do an MRI for brain, we do brain whole body, and in association with corneal confocal microscopy and vibration sensory tests, especially for diabetes. Then we have also associated with a cognitive test, we use Cantab test for attention, psychomotor speed, executive function, and episodic memory. So for the blood, as I mentioned, that we collect blood, urine, saliva, and stool now, which is recently done. For the blood, half of the blood we store it in the biobank, and the other half we send it to the hospital, where we collect 77 biomarkers for all our participants, the baseline and the follow-up. So it is divided according to systematic approach for them. So I will not detail this. Then this data from the hospital will return back to our participant. The main attraction for the participant to the biobank that after doing all of these tests, we provide these results to the participant. We appoint for them another appointment after 2 weeks or 3 weeks, depending on the availability. Our medics team, they sit, review with the participant all their results, and refer them in case they are referral. So we have to be connected also to the hospital and the primary health care according to the request of the preference of the participant. So it's very important that we have data integrity, and we need to validate our data continuously because we are connected to different resources. So as mentioned, it is basic that the foundation of any research, that the accuracy and consistency of this data is very important, and also support to precision health due to accuracy by providing accurate data and personalized treatment. Then we have to keep the trust, either the trust of the participant to come, and trust of the decision maker, and also and regulatory compliance, and also trust of the physician, the referral, that we are referring the hospital and the primary care. So what is the approach we took it in the biobank? We did start with the data harmonization, as we are receiving the data, also electronic medical records. We have more than 15,000 electronic medical records, so we have to detect the mismatching between the sample collected and also handling. We have to continue quality improvement. And of course, data integration and data verification from all the process, as we do DNA extraction, RNA extraction within our premises. And for sure, we have to keep the data security and privacy. So for that, we did data standardization. We have to follow uniform format and across the database, which we have set in the biobank. And we ISO standards, we are ISO certified for QMS, ISMS, and also for biobanking. It's very important that we have to be accurate numerical precision for the accurate data, per time measurements between different rooms, and all of these steps for validation. And of course, to control, this is the SNOMED system. So as I mentioned, that we have a plenty and wealth of data, and it's need to be received from the hospital as we send the samples, and they return to us. We have also service providers that, especially for disease base, where they select the cohort, the researchers from the hospital, and we share the data with them. We have, of course, our customers, and on-premises system, the core laboratory, which is we are connected, and through cloud system. So as a challenges, which is very important, that as I mentioned, I'm concentrating here on the electronic medical records, because when we received, we thought that it's simple work. But unfortunately, it was so difficult that we found that the electronic data, it changed from within the hospital. So we have SNOMED data, we have ICD-10 code data, we have CPT4, and also PNED data. So our scientists sat together, so we identified to do harmonization, because we want to provide the researchers with the same standard data and selected that Australian modification, which will be standardized, so work on this harmonization of the data. For another example, that between the IDEXA, as I mentioned, that we refer our participant to the hospital, and we are research center, like we are doing the IDEXA. We found that more than 40% of the participants that we have vitamin D deficiency, and we have osteopenia and osteoporosis. So for osteoporosis, they have to be referred emergency to the specified clinic at the hospital. Then once we refer the participant with osteoporosis, they reply back, because as a continuous checkup for the quality, we receive a report about the accuracy of our data from them, and we said they said, no, it is not osteoporosis, it's osteopenia. But when we have a meeting with them, we found that they are using different references, and they are convinced at the end with our results is more, and they change and they accept the participant about that. So it is T-score is calculated according to different calculations. So the population calculation is our calculation was the better. So this is the way we overcome this data, and especially if they want to recruit participants and establish another study related to the bone density. So for mismatching detection and handling approach, we have to do cross-validation. As I mentioned, we have data profiling and manual review and intervention. Especially as an example, we have in Qatar that we are using the registration with QID, which Qatar identifier, this is a number, and the hospitals, they have another number, which is called HIC. So sometimes from the beginning, from few years ago, you will find that the two numbers are not giving you, we have to register because we are referring the samples to the hospital. So they have to go together, and the numbers become correlated together, and we find mismatching between them, and even the names, because the names are common family names in there, so you'll find the challenge is that, but we don't use a name. Now we use the date of birth. So we decided that as an identifier using the QID and HC number, and contacted the hospital to be sure about the correlation between them, and use the date of birth as a third identifier. So this is very important for us as the data from participant, self-reporting data. This is another level of cross-validation that we ask a question about if they are diabetic or not. Then we measure HbA1c, or doctor asking within the questionnaire itself, we do a cross-validation. So example for some people, they don't know that they have diabetes, but sometimes they mention that they are well, and they went for the doctor and they don't have anything. So there is a discrepancy between their questionnaire and chronic disease. Once we find that they're even look to their medical records, there is a discrepancy which is around 9%. We are working to avoid that. So we have to be sure or highlight those data which we discover that there is a discrepancy. So this is very important, and also that identified discrepancy, as I mentioned, between the different codes. Now, another example with the MRI. We use the manual review. Like when we have the MRI, we agreed, we contracted a radiologist from the Hamad to review the quality of the MRI and put a report. So we have a report about the brain and another report about the whole body. So this is very important that to feel and review directly about the quality of the images which we have. So in general, for MRI brain and the review from the radiologist, that we have around 96% of 97% with good quality, and we have 3 with blurred image is that the radiographer, they didn't do a good job at the time. And for the whole body, it is most of it is like 99.5% is a good quality. So for the data, structured data, as I mentioned that, we have the data is important that it's kept in fully automated, as I mentioned, with ICD. This is our model and our future collaboration to automate everything, like to use the AI model for fully automation, not depending on the scientist for transferring the code to different ICD diagnostic code from patient documents directly. And especially the information we find, that most of the information is written as a note from the doctors. Sometimes they are simply entering any disease they find, they don't go with accuracy like A, and they select whatever first thing, they are so busy. So that's why, and in the note, which is not transfer, we have to work on it with the AI to be transferred. And as working in close collaboration with a hospital and continue training to improve the model with new data. This is our objective now, and the project is working on for easing the integration. So this is the important, as I mentioned, is all of this to improve the accuracy of the data and increase the volume and increase the revenue. For the data security, is very important to attract and keep the participant that for confidentiality and integrity of availability of the data and the data privacy we are following. In Qatar, we have a data privacy law and we have data protection principles, as I mentioned, and access control for the researchers. And as I mentioned, we are ISMS certified for data. So for precision health as prevention is very important that to close the loop, as precision health is started from prevention, precision health medicine, and precision health care. So to protect and return the value to patients through transparency, engagement, and storage focus on data privacy. Strengthening of participant-scientist relationship, it's enhanced genetic counseling program, because we do now, this is the idea, the project, the successful example. As I mentioned that we have like now around 30,000 participants, we did for them a whole genome sequence. So from the first 6,000, there is a study that it was that they will find on about the BRCA1, BRCA2 for cancer prevalence within the first 6,000 participants. So it was at the beginning, it is a research study. So we concentrated on BRCA1, BRCA2. So we need to validate those participants, like around 28 participants, they found them, and to active or to intervene and what to do about them. So this is the successful story who was working with Hamad Medical, with the cancer hospital at HMC. So that we started the study, we have to recall the participant, reconsent them if they want to do, to confirm the BRCA1, BRCA2, and then once we send the samples to the hospital as it is certified lab, then we return the results. We found that it is all the participant, which they signed the consent that the results of the research, it is the same as the results of diagnostic one. So then we as the new consent, we refer them to the specialized clinic, and the specialized clinic, they follow them and their family. And it was successful because most of them, like around 14 participants, were male. We were so worried about the accuracy of the data. Is it there is any mismatching through the way or something, because we didn't expect that. But it's proved that the study, one of the implementation study through a research project. So this is a very important that we leverage the Biobank model in personalized healthcare. So at the end, I would thank you all for your listen and thank my colleague. And unfortunately, she didn't able to present this. And I hope I covered the main points for you. Thank you.
Thank you very much, Professor Afifi. So our next speaker is Professor Andrew Morris from Health Data Research UK, who'll be speaking about gearing an entire country for precision medicine. Thank you.
Thank you, Sarah. What a great pleasure to be back in one of the world's greatest cities. It's almost as good as Edinburgh, I should say. But I think you'd agree this precise IHCC symposium, arguably, to my mind, has been the best international symposium, which has showcased the world's global package of cohorts. It's just wonderful. But what I'm going to try and convince you over the next 20 minutes is that as a community, as a team, we have an opportunity to leverage even greater value from these cohorts. But to do that, we need to embed them in national health-relevant data infrastructures to see a convergence of care and research. And if we're going to deliver on Jeff's vision of making cohorts the future hubs of innovation, then we need to see this convergence of care and research. And I'll canter through this because we need loads of time for discussion. So I'm going to talk about the pace of change, how we're trying to do this, and we're not there yet in the UK. How are we gearing an entire country for healthcare research and innovation? We're using data science as the catalyst for change. And the hypothesis is that health-relevant data should be seen as critical national infrastructure. And it's time to be bold, ambitious, and enterprising. Pace of change. I was born in 1964. I'm an old guy. And there were only 3.4 billion people on the planet. As you know, November 2022, UN suggested there were 8 billion people. How are we as a community going to deliver care, equitable care for 8 billion people? We need to innovate. Inequalities has been quite rightly a theme of this meeting. This is a picture of the UK inequalities. And we've coined a phrase, coastal health, because inequalities in the UK are not in London, Oxford, and Cambridge. They're distributed around the country. So how do we ensure that what we do is representative? And pace of change, and I'm a doctor, but I sort of fell into data about 20 years ago. And in my world of diabetes, we've seen massive changes in GLP-1 agonists, insulin pumps. But in the world of computation, there's been an equally rapid pace of change, whether it's sensors, robotics, natural language processing, and we're also seeing a confluence of these technologies. And the future of medicine is at the interface of math, statistics, computational science, and domain, and that's our world. An example, by 2030, there will be 30 billion ubiquitous devices connected to the Internet of Things. And to illustrate this is a picture of one of our very many recent prime ministers looking around the supercomputer. And the UK is currently considering whether we should invest in exascale, which is 1 billion computations per second. It's unimaginable. And we're seeing a rapid progress in AI driven by compute and data. What does AI need? It needs data, compute, and people. And what we're now seeing is that AI has surpassed us, humans, in delivering tasks, whether it's handwriting recognition, speech recognition, image recognition. But in recent years, as you can see, the rate of progress is just remarkable. And data are the feedstock of AI. So let's not get misty-eyed about the algorithms. Let's think about the data and the quality of the data. And I would argue it's going to become increasingly important. So this was in Nature 2 weeks ago. And there's this concept of AI collapse. An AI collapse is that when you retrain and retrain on increasingly synthetic data, the original content distribution curve disappears. So what we're going to need is increasingly high-quality human data. And that's why the cohort community is in pole position. So I got my head around that paper, but I thought it was really well illustrated by that. So that is how you retrain on common pedigree dogs. I must say that's how I used to look on a Saturday night when I was a student. So, and Lloyd Minor, who's the Dean of Stanford, said, why AI is medicine's biggest moment since antibiotics. So let's think about the data. And this community has a great opportunity to shape the future of medical science, because that is how data looks. As we've just heard eloquently from Qatar, there's a lot to do to engineer and sort the data. Who here was frustrated with the 3-pin plug in Singapore? Who forgot their adapter? And the other thing which we've heard is the importance of scale. So we've heard about UK Biobank. Framingham, we heard from Jeff yesterday. So that's the relationship between blood pressure and mortality from Framingham. 5,000 people, 50,000 people, 500,000 people. So scale is going to be increasingly important. And where are we with AI and adoption today? Health is seen in green here. This is some work from Aradazi from the WISH consortium. So we're sort of mid-table at the moment. You know, we're not out there. Financial services and high tech, so mid-table. In the UK, we say that's like Manchester United. So what we wanted to do is collectively do the heavy lifting once, the pre-competitive stuff, infrastructure, data, and services to sort the data. So 6 years ago, a consortium of funders, I thought it was prescient, got together and said, let's try and do this once, because other industries, whether it's banking, telecommunications, airlines, they have a way of messaging, and they usually have a neutral convener who ensures the rules of the game. So we established a national institute, and we said, we want to unite the UK's health data in all its relevant forms to enable discoveries that improve people's lives in anticipation of the fourth industrial revolution. Sort the data on 68 million people. So what we did is we said, right, let's have team science across the UK, and these are our fantastic research leaders whom I hang on to their coattails, but around a single mission to sort the data. And our vision is to create an e-cohort of 68 million people. So everybody in the UK. And this is the classical Framingham slide. You have diversity in detail across the lifespan, exposure, outcomes on 67 million people. And my hypothesis is that we'll see a sector maturity. We were late to come to data ecosystems. Classically, I'd phone up my mate and say, do you have data? And you'd have a bilateral relationship. But what we'll see is a democratization towards fair data so that we'll have many-to-many trustworthy relationships. But to do that, you need data, connectivity, governance, trustworthiness, benefit sharing. It's the engineering of the data ecosystem. So I said, when we first started, I said, let's map the system. And then the team came up with this graph. I said, oh dear. But I would argue it's the same in your country. It'll be the same in your country. And indeed, I went to Holland recently, and they're the same. So it's this, how do we engineer the messaging across a complex data ecosystem? To do that, we have an institute without walls. The easiest thing would have been to build a building in London or on the Euston Road. But we are an institute without walls. We're independent, so we sit in Wellcome, in the Wellcome Trust. Because if we tried to do this from an individual university, it would not have worked. We've got a strong independent board, and our job is to try and drive standardization, harmonization, infrastructure, data services, access, governance at scale. And these are just some of our board members. So our triple aim is threefold. How do we sort the data to accelerate trustworthy data use? Secondly, and we borrowed this from the Global Alliance, how do we get driver programs which show the art of the possible? And thirdly, this is about team science. How do we shape the future of health data research in terms of an equitable approach and democratize access? And can I just make a shout out for data engineering? Because we spend far too little time on data finding the data, accessing data, linkage is key, curation, automated curation. You need to do that pipeline before the data is useful. And that's the expertise we're trying to develop and celebrate. The other thing is you need a data strategy. So this is our national data strategy. We have national data, we've got the sort of what I call regional EHR data, whether it's lab, imaging, text. We then have health relevant data, so that's stuff like education. It's stuff about employment. We've got research-generated data. You've heard a lot about Our Future Health and Biobank. Environmental data, but also increasingly personal monitoring data. So how do you get your arms around a data strategy? And key to this is to have an engineered, scalable, reliable, sustainable, secure, interoperable platform, which is not only the tech, it's the governance, and we really like the 5 safes model: safe people, safe projects, safe places, safe data, safe outputs, and drive interoperability so that we connect consumers, researchers, innovators, analysts to content providers. And it's actually, in the early days, we went to see Amazon. And Amazon is a scalable model of connecting content providers to consumers. And I mentioned driver projects, and we stole this from the Global Alliance for Genomics Health. So these are projects which do 2 things. Firstly, they do great science at scale that individual universities can't do themselves, and they clean the pipes of the infrastructure. So they leave a legacy benefit, whether that's for governance, data, etc. So these are this handful. We've got one on prescribing. So we've got nationwide prescribing on 68 million people. John Dinesh, Cambridge, Sarah Lewington, lead one on multiomics, which is linking 14 multiomics cohorts to the electronic health records for 750,000 people, bringing together data such as the Olink platform and others. So we've got these drive codes. So how's it going? This is not Scotland, I should say. This is a base camp of Everest, because we are in the foothills of where we need to be. First of all, this is less about tech. This is a people project. And in the early days, we worked with Sue's colleagues at NHS England to say, let's create an alliance, again, modeled on the Global Alliance. And we've got 110 increasing data controllers who all subscribe to this ecosystem to make their data fair across the UK. In terms of productivity, we want to make research easy for folks so that people with the best ideas can access their data. So in the first 5 years, there's been about 4,000 papers, 200,000 citations. But importantly, this is a measure of collaboration across the UK. And it's enabled analyses at scale. So this is a study on 58 million people. Cross-sectoral data linkage, health and education, personal monitoring, because it's a scalable model. Now, key to this, and we've heard a little bit about this, is trusted research environments. So these are, it's like a reading library, so that subscribing to the 5 safes, people, projects, setting, data, and outputs, people can find data and then run their projects in multi-tenant, cloud-enabled analytical platforms. So we don't need 100 of these in the UK, but the phrase I use is power stations on a grid. Power stations on a grid. And subsequently, the NHS is investing in a national trusted research environment and 11 regional ones. Population denominator about 5 to 7 million for those high-resolution intensity data sets. So a national architecture of infrastructure. We then put a fabric over the top of it, and anyone in the world can search the metadata of UK data. There's about 900 data sets here, so you can find data and then request access to it. And to give you very briefly a case study around COVID, this is our colleague Patrick Vallance, who's now actually a science minister. He was the chief scientific advisor. During COVID, he said, what are those things we should do as a nation So we looked at trials, transmission environment, etc. But he also said, how do we connect this together on a UK-wide basis? So he invited us to say, right, connect the data, map the data, develop and enable the infrastructure, and then make the data available so that we could ask questions during the pandemic response. And we, again, pulled the UK together. So these are the national sort of trusted research environments. This is a busy slide, but it shows vertically Northern Ireland, Wales, Scotland, and England. That was the data estate fairly early on in the pandemic, and that's what it was 14 months later, the ability to link across these health-relevant data sets. What can you do with it? So this is Aziz, who led the first ever study demonstrating the effectiveness of single-dose BioNTech and AZ vaccines. And it was a great platform as a service in terms of real-world vaccine effectiveness, variants, vaccine waning, etc. So it's very powerful. Now, why can't we do this for cancer, cardiovascular disease, mental health? It's the same model. And then, likewise, with NHS England, the colleagues, we were able to look at data at scale on 56 million people, looking at the vascular risk of various vaccines. Remember VIT with the AZ vaccine, really nailing down the absolute risk. This was looking at long COVID. This is Jonathan and Angela. Jonathan from Bristol, Angela from Cambridge. How long is long in long COVID? So I phoned them up. I said, that's a fantastic paper. And I knew it was good because it made The Sun, which is our very popular UK newspaper. Urgent warning to anyone who's had COVID in the last year over killer complication. So, and then it enabled trials. This is Martin, Peter, and Marion, the recovery trial. The key is data-driven innovation to drive feasibility, eligibility, recruitment, and ascertainment of outcomes of trials. And the beauty of the recovery trial was the outcome data were all derived from routine data sets. So it really drove the efficiency of the study. And I think we heard yesterday, this really put our world high up in the news agenda internationally in terms of people understanding the value of data to drive improvements of their care. And then this year, for the first time ever, we published on everyone in the UK. So this was called the Coalesce study, again, a partnership across the UK. It was a simple question. It was looking at antigen status. It was looking at outcomes and under-vaccination. And it was able to show the disparity. But key to this was that we can now run studies on a UK basis. Internationally, and we were privileged to work with Nikki and colleagues in the pandemic, we worked with the Gates Foundation and Minderoo from Australia, because there was a need to support and work in partnership with colleagues internationally and provide governance, infrastructure, data, and services to actually run studies across, well, we ran studies across 60 countries with 12 driver projects. And this was really global team science. And we sort of reviewed it recently in The Lancet. Here was an example, resilience of health systems in 10 countries, because this infrastructure is scalable. Of course, that's mostly structured data, just to focus on unstructured data. Sarah's hurrying me up, so I'll speed up, Sarah. So this is Pierce from Moorfields. We've mobilized 1.4 million OCT scans, that's optical coherence tomography, and then linked using generative models to look at incident disease through linkage 7 years later. And this stuff was in Nature, showing you could do an eye scan and actually predict incident Parkinson's 7 years in the future. And I must speak to the Enigma team, because we're beginning to curate national imaging archives. This is the Scottish example. We've got 1.6 million brain images within a supercomputer environment, and we're now analyzing this for biomarkers of dementia, motor neuron disease, and diagnosis. And I'll skip through that, gives you an idea. And finally, I think our cohort community, a challenge for us is how do we enable trials? And this is an example. We're working with Sarah and the team at Dementia Platforms UK. Can we take our enriched trials population, create a risk prediction engine using blood-based biomarkers, genetic biomarkers, and blood biomarkers for dementia, and create pre-screened consented cohorts that will really drive clinical trials and the efficiency of clinical trials. So this is something that we're currently working in partnership. There's a whole pipeline of drugs for neurodegeneration, disease modification, but we need far better ways of targeting those to patients in need. Currently, the failure rate in dementia trials is 79 patients out of 80 for recruitment. But we're seeing blood-based biomarkers such as p-tau, which can actually enrich. So how cohorts enable trials. So to summarize, my challenge, can we as a community sort the data? And I would argue it improves care, it drives population stratification, it optimizes trials, it manages the health service, and it drives research. It's the same script. So we need this convergence of care and research. To do that is team science, and we need more computational scientists, ethicists, lawyers, data engineers that we can learn from to deliver this. And I'd ask us as a community, how do we systematically drive a convergence of care and research from cohorts to clinics? Because otherwise, research will be out here, it will be difficult to fund, care will be here. The beauty of Sue's presentation is we've seen that convergence of research and care. So how do we do that at pace? And I've always had, it was through Steve Kern at Gates, can we envisage an international mesh network where we can run experiments across global health data science capabilities, which actually will allow us to enable science that currently is unimaginable. And I do think Singapore is leading the way. They've got many of the components, Precise, the SG100K, Trust, the National Cohorts Office. This is from the Straits Times yesterday. Family doctors can become specialists. They're investing in that health data infrastructure. So to conclude, we're not there yet. One, this is Marie Curie. One never notices what has been done. One can only see what remains to be done. I do think that the new Einsteins will be the scientists who share. So all credit to this community because you're a sharing community. And finally, as we've heard, there will be bumps on the road. So when we set up HDR UK, I said, we need a website. So the team went away and they said, it's tricky, Andrew, because Havering Demolition and Recycling have got the HDR URL. So I thought, and I didn't think this would take us that far. So we used a little bit of MRC money to buy them off. So can I thank you for listening?
Thank you very much. And can I just invite all speakers up onto the stage? And just a reminder that after the tea break, there are 2 parallel tracks on population health and genomic science in cohorts. Please do submit your questions on through the Pigeonhole QR code. And also, Mr. Peter Goodhand, co-chair of the IHCC, will also deliver his closing address and announce the winners of the poster sessions after later today. So for now, we're going to have a Q&A. Okay, well, thank you everyone for submitting your questions. There's some really good questions come through for all of our speakers. So I will try to balance them across the speakers. So being Andrew's just gone, we'll challenge him first. So one question here, is it coming up? No, I have to press harder. It's not coming up, so I'll just read it. With HDR UK, how does it work with UK Biobank environment? And is there a plan to unite towards improving the health outcomes? And by the way, great talk.
Thank you. Good question. So what we'll see, I think, in most countries of the world are what I call large program-specific health data infrastructures. So in the UK, we've got 2 or 3, for example. Genomics England is a large data repository. Dementia Platforms UK is another one. Biobank and Our Future Health. And these are great assets in their own right. But what we're trying to do is to enable linkage of those assets to all the other data that I showed. So our role is to drive interoperability, support linkage to data which aren't collected by Biobank. For example, can we support Biobank to link their data to air pollution data, meteorological data, or the multiple health-relevant data sets which sit across the 4 nations of the UK. So it's that linkage. But that's why I quite like the phrase power stations on a grid. We provide the grid capability to provide an oversight of a unified UK system. But we're not there yet, but it's the sort of architecture.
Thank you. Yes, I've been asked, is there one platform better than another, and why should we use yours? And I think you've just answered that. It's more about everyone working together to deliver a cohesive benefit. A question for, well, this is a general question, actually. And with more cohort data, there'll be more rare variants with a severe lack of information. Let me just try and click on, you see, I'm trying to hold too many things with my hands. That's why I can't click on it. Let's click active. Is that up? Yes. There will be more rare variants with a severe lack of information on their effect and pathogenicity. How do you overcome this to truly identify if they're clinically significant? Which of you is this more relevant to? Perhaps Nikki?
So I'm just a data person, but I did come from the lab, and I think it depends on how urgent it is. You know, the need to answer the question. You need functional studies. I think functional studies in mice, for example, can maybe help. But when it comes down to sort of making a decision much quicker than that, I think you just need to get all available data. There are a lot of tools that now look at protein structures to see, obviously, if it's coding variant. For non-coding variants, I think you need to obviously start looking more widely at expression data, methylation data, epigenetic data, and I think we should use AI more. But of course, dirty data gives you dirty results, so we need to make sure that the data that everybody is cleaning and curating actually becomes available to train the models, and it has to be context dependent. It has to be relevant to the context. So I think it depends very much on the urgency, but functional studies and deep sort of molecular analysis, I think, is the number one choice.
Yeah. Indeed, thank you. That's it. So I'm going to move swiftly on because our Q&A time has been cut a little short. Oh, no, I'm doing really well today, aren't I? Let's see if we can get this question up for Dr. Afifi. How do you address or justify the limitations set by your country's data privacy laws?
In Qatar, we don't have a limitation. We have a law, which is GDPR, following the same is published, that for transferring the data, controlling the data, and especially even for storage the data on the cloud. So there is a privacy law in Qatar. So, which sometimes it will cause a limitation of changing or between different countries, but there is an approvals and exception from the ministry about in case that there is a committee for reviewing all the international collaboration to approve that. So there is a law, but the law will not prevent the collaboration.
Great. That's, I think, a good answer. So we have a general question here. What is the acceptance rate to be involved in given population biobanks? Any comment from any of the panel?
So from our side, to be accepted, it has to be adult, and this is a control for our population, for the population by study, that they have to be adult and long residents, either Qatari or long resident who stay in Qatar more than 15 years, to ensure that the follow-up data is there. It will be there. But people, as I mentioned, that the local are small amount, and most of the country are expats who leave soon. So the data will not worthy after a while. So this is the main for the study. For the other disease base, it is from the hospital as a patient. They will be recruited and follow up according to the study itself.
Great, thank you. Andrew?
It's not my specific area, but I understand UK Biobank successful recruitment was about 6 or 7%. So 7% of folk who were invited participate. I think our Future Health's better than that, but I think it's about 10, 14%. So that's why representativeness is so important. And we heard a fantastic example from Finland yesterday. If you've got data on everyone, you can actually look at the representativeness of the cohort of interest in terms of population data. So it's about 6 to 10% uptake rate. So it's quite, you know, people forget that, I think, about Biobank.
Yeah, thank you for those comments. Right, the next one is, in terms of validation, the findings in one specific cohort with a specific race, such as Asian cohorts, is it recommended to validate the findings in other different cohorts, such as Caucasian population? Anyone like to answer that? Yeah,
so a lot of the Asia Africa studies actually did try to do validation in other cohorts, and actually it was usually European because that's where there was data. But I think it's less relevant, and you know, many of the times it was a different variant. But often the same gene or the same pathway, but a different variant. So I think it is important to validate, but preferably in a population that's relevant to what you're trying to do. I think it's the question you're trying to answer. You know, are you trying to see is it global, or are you trying to see whether it's relevant to your population? So I think that should determine which population group you choose to do the validation in.
Great, so it's more strategic than just let's find another population with a good number. Yeah. Okay, so let's have a look here at this one coming up. How do we assure health equity in AI models? Who's going to take that?
Sure, there's more data from the underrepresented data groups. I think that's and skills. So we need skills. We need to make sure that there are skills in all countries, because it's something that you can do without a lot of money. You know, you don't need big equipment, you know, to generate data when you like you would to generate data to analyze that data. So I think the equity is both in what is the health, what is the data, is it representative, and are there skills across the board to analyze that data?
So here's a question from me off the back of that one. Do you think we have adequate training for the next generation of researchers, which is affordable across LMIC and other income countries, to actually deliver on these AI models adequately? Or is this a gap that needs to be resourced?
I cannot speak because I can speak both of them from Qatar and Egypt, because they are different, rich country and low-income country or middle-income country. There are people also, they are graduates, but they need the support for their specialty. They need more training more collaboration, more exposure to what we are doing here, even in the conference and all of that. So I am sure that there is an opportunity for future, yeah.
Great. Andrew?
So a few comments. Firstly, I think traditionally academia has completely undervalued the really talented people who are data managers, data scientists, and their career path is not as well defined as it should be. We need to celebrate, support these people and put a fair wind behind them. And often they are remunerated different. They're on an administrative pay scale rather than so I think there's a big issue there. Secondly, around the role of industry in this, because what we're seeing, and it's acute in some of the big centers like Boston, London, is that as soon as people graduate with a PhD or master's, industry suck them up. So I think in the university public sector, we need to be really smart about how we provide people with a rewarding career pathway, because otherwise it will all be sequestered by industry, which I don't think is a good thing.
No, I couldn't agree with you more. So the next question is for Nikki. What are the concerns for federated analyses with Nomad? Will algorithmic innovations that don't need sharing of individual level data make a difference in your case?
Yeah, so we're actually working with Nomad for this federated Nomad approach that Heidi talked about. But the federated analysis that we were working on is when you're trying to do joint calling across data sets and you don't want to have the individual level data there. So that I think is where the biggest value is, because when you've got summary data that is much less identifiable, it's much easier to put that in a more central place and compute on that. But there are a lot of challenges, if you're trying to do joint calling, and technically data sets need to be next to each other. And I think federated analysis is the best solution for when you do have to worry about making individual level data available, and making sure, and also algorithms that can work on encrypted data. So not even the algorithm sees the data unencrypted. I think that'll even be even better scenario, because then it really is truly protected in terms of the individual level data.
Yeah. Just to add to that, another of my questions is through federated analyses, we're trying to do this ourselves in Deep UK at the moment. Do you think that there is a risk of diluting the analytical capability? So rather than a very complex model, a more simplified model that can be enabled through federation, do you think that could be a limitation?
Yeah, absolutely. I think you always have to simplify things for federated, until we get more technically advanced.
Yeah.
I agree. Yes, for us, for individual level data, it will need a special approval for that in case, because at the end of it is referred to the certain participant who are within the biobank and participated and returning. So there is a way, but in general, we don't allow downloading the individual level data except with special approval from the Access Committee.
Yeah. Thank you. Okay, the next question is for Dr. Afifi. Are there challenges related to having blurred lines between doing research and using the results and data as clinical information while collecting data for Qatar Biobank?
I think the blurred line is within MRI, because we have like 4 MRI technologists. They are taking the care of taking the images, and so we have to follow the performance of our staff. So that's why, in general, it's the performance and giving them training later on and identifying where is the lack for that. So the quality of, we accept like less than 5% if they are not covering, and we replace, like if we receive any comment from the researchers, they are not happy with this, we can replace them with a new MRI. So this is how we are trying to overcome this challenge which we have here.
Thank you, thank you. So the next question is one that actually I have. You've all spoken about this on data standardization, metadata, harmonization. With the complexities, in DPUK we've developed an ontology, but do you think that actually it will become country then domain specific for metadata curation standardization rather than a one size fits all?
Yeah, certainly in the DSI consortium, we've realized that you need different data models. You know, we use OMOP for clinical data. The climate change people have their own models. So we even before we started, we decided that there's going to be different models for each domain. Otherwise, it's just going to be too much of a blur, and you're actually going to lose data if you try to map to something, you know, one single model.
Yeah.
Yes, I agree that within the biobank, of course, we will have our own catalog for the data which we are collecting. In spite that, as I mentioned, that we have a unified electronic medical record, which is cerner within the country, even the private hospital and all of that using, but we find that there is two versions. Even CIDRA is using one version, and for the other hospital using another one. So this unified, hoping it will be successful at the end of the day, to support everyone here.
Sadly, there's no lack of standards. It's a question of adoption and enforcement. So I do think speaking with the vendors to try and standardize as much as possible. I also anticipate major developments. We're seeing lots of SMEs being set up around AI for data curation, so that it will, because at the moment it's very manual. So I think looking at automating the mundane using AI for data curation is an area that I think will be increasingly important.
Yeah, thank you. Well, thank you panel. Thank you for your presentations and the Q&A, and thank you audience for your participation as well. Thank you.